As the Executive Director of the Basser Center, it's really been my privilege to witness firsthand what's been happening in the field and the role that the Basser Center has played in all of this. We know that there are just ongoing global collaborations which bring together researchers from across Penn, a nation in the world, to share innovation, to share knowledge, and we are excited to help lead all of these efforts. None of this work would be possible without the Basser Center and the incredible generosity of Mindy and John Gray, who founded the Basser Center more than 8 years ago and whose vision and leadership continues to guide us in all we do and encourage us to continue to do better. We are grateful for all of you tonight who have been part of this collaborative effort. It takes advocates, genetic counselors, physicians, philanthropists, everyone working together to really make a difference in this field. We are proud of what we have done, but already you can see as we talk today that there's much more work to be done. I'm watching the questions in the comment field, and so many of the questions are how we can move from surgical prevention techniques to things that are better. In the midst of this incredible pandemic, the last 6-7 months, how long has it been during COVID-19? We've all continued to do work. Some of the laboratory work was placed on hold, but it's back again. And we will get through COVID-19 and we will continue to focus on what we need to do for BRCA1 and 2 and for all the families that are dealing with these difficult issues. For all the families dealing with BRCA1 and 2 mutations, we can and we must do better. We must give better choices to those that are newly found to have gene mutations. We need to give better therapeutic choices for those who are dealing with cancer. So with all this in mind, it's really my pleasure to introduce two incredible advocates of the Basser Center, Jerry and Len Potter, who established and endowed the Basser Global Prize to honor a visionary scientist who had advanced BRCA1 and BRCA2 related research that's led to improvements in clinical care. So I'm going to pass over the baton, if you will, to Sherry and Len for their comments. Thank you so much.
Thank you, Susan. This year, more than ever, we see the importance and the power of scientific research. We all feel the urgency and the magnitude of a push to find a cure or a treatment. Well, this is how people with BRCA mutations feel all of the time. We're always looking for better screening techniques, better treatments, and dare we say a cure to the cancers caused by this mutation. That's why it's truly an honor to be a part of the Basser Center and this symposium, and to witness the passion and intellect of some of the world's most brilliant minds coming together to change lives and alleviate our fears. Though we wish we could all be together in person, we're glad that you'll still be able to share and learn from the research and clinical ideas and information that flow from this virtual symposium. Len and I established the Basser Global Prize in 2013 to help foster new ideas. The prize awards a significant, unrestricted, unconstrained grant to pursue innovative BRCA-related research efforts to a leader in the field. The goal was to provide brilliant scientists the resources they need to pursue promising new research that'll lead to innovative new approaches to cancer treatment and care. And over the last 7 years, the prize has done just that, providing grants to fund projects that'll make a difference for individuals and families with BRCA1 and 2 mutations, giving them better options for prevention, targeted therapies, and improved quality of life.
This year, in a first for the Basser Center, we honor not one, but two of those brilliant researchers, Drs. Douglas Easton and Antonis Antoniou. Dr. Easton is a professor of genetic epidemiology at the University of Cambridge, and Dr. Antoniou is a professor of cancer risk prediction, also at the University of Cambridge. Their work, which focuses on genetic susceptibility to common cancers, with a specific focus on characterizing the cancer risks for BRCA1 and BRCA2 mutations, has significantly advanced our knowledge of genetics and cancer risk prediction. Their ongoing efforts may help us better understand who bears the greatest risk, information that has the potential to truly be life-saving. The prize that we award today is a replica of the large sculpture hanging in the lobby of the Perelman Center for Advanced Medicine at Penn. This sculpture, called Homologous Hope, was created especially for the Basser Center by internationally renowned artist Mara Hazeltine, and is an accurate depiction of a part of the BRCA2 protein that is involved with DNA repair. The light from this prize symbolizes the hope that you and the Basser Center are giving to countless families and their loved ones. Drs. Easton and Antoniou, we are grateful for your work and hope that the prize provides you with the financial resources to continue your research and to allow your genius, your creativity, and your inspiration to continue to flourish in support of our collective mission. It is your effort and your dedication that moves us closer to our goal of ending BRCA-related cancers. Along with Dr. Domchek, we are honored to have you with us and to introduce you. We look forward to your keynote presentation.
Okay, thank you very much. So it's a tremendous honor to be receiving this award together with Antonis, my long-term colleague and collaborator. I'd just like to start by thanking the foundation, Susan and all the organizers, Barry and Len, and Mindy and John for their tremendous efforts in supporting BRCA research and their generosity and drive and vision, which is making a tremendous difference to the area. So when I started to prepare this talk, I was rather horrified to realize that actually I've been working in this area for more than 30 years. And so I'm going to take a little bit of a trip down memory lane. And when I started back in the late 80s, it was a very, very different world, of course. There was no clinical cancer genetics to speak of at all, really. But a number of groups were getting interested in trying to understand genetic susceptibility to common cancers, which clearly existed. We didn't have very much technology, and so a lot of time was spent collecting families, like this family shown here, which is from Ireland, where you've got a number of individuals with breast cancer, usually diagnosed at an early age, ovarian cancer you can see, pancreatic cancer, male breast cancer, and so forth. And it certainly looked like families like this must have an inherited basis, but to begin with, you didn't really have much of a way of getting a handle on it. Our main technique at the time was to use the now being developed polymorphic markers, so-called microsatellite markers, these repeat markers that you could use to try and track chromosomal regions through families. It was pretty rudimentary in a way. These were markers that you had to read off autoradiographs, and we spent a lot of time trying to stare at them, trying to work out what was going on. But one thing which became very clear was that this wasn't something that one group was really going to be successful at. And we needed a wider collaboration. That's really been a theme over the last decades. So we got together with a number of people. Some of the key movers are shown here to form the Breast Cancer Linkage Consortium. And incidentally, the picture on the left there, which is a younger me together with David Golgar, who many people know from Utah, that was taken in an Irish pub in Dublin. You see David holding a pint of Guinness. And in a way, one of the main themes was the ability to meet together and discuss new ideas. It's rather poignant that we're having this presentation at a time when we can't do that. Anyway, through the consortium, we were able to gather together collectively a much more informative set of families, including families like this one, which is a rather famous family from Utah, which was originally studied by Gardner in the 50s. And this family, together with the Irish family I just showed earlier, were really the the key to identifying BRCA2 at the beginning. So the mapping of BRCA2 is illustrated here. So we're back to this Irish family, and here we see a whole series of a haplotype of markers on chromosome 13, illustrated by this bar. And you'll see that, if you look carefully, that all, or at least most of the affected individuals in this family carry the same haplotype. So that together with the evidence from the Utah family and a number of others, showed definitively that there was a breast cancer locus on chromosome 13 in this region. And indeed, it turned out that this family has a nonsense mutation, a protein truncating mutation that was discovered a year later. So a year later, in 1995, the whole region was sequenced, and it could be shown that a number of families did indeed have protein truncating mutations in this gene. So this was the discovery of BRCA2, and alongside the discovery of BRCA1, which was a year earlier, we now have 2 high-risk breast cancer genes. So one of the main things which the BCLC was able to do was to document the contribution of BRCA1 and BRCA2 to families with breast or breast and ovarian cancer, and a very clear pattern emerged. So it became clear. So this was studies with now several hundred families across the world, and it became clear that most families, almost all the families that had breast and ovarian cancer, either had a mutation in BRCA1 or a mutation in BRCA2. Families with a male breast cancer in them usually had a mutation in BRCA2, but families which only had female breast cancer was a more mixed bag. So a significant fraction, actually more than half, didn't have a mutation in either gene. And these became known as the BRCA-X families. And I think it's fair to say at the time that everyone was assuming that if we just did more linkage studies on these families, we would soon enough find the BRCA3, 4, 5, and so forth. But unfortunately, it didn't quite work out like that. At the same time, a couple of large population-based studies were initiated, which did sequencing of BRCA1 and 2 in unselected cases of breast cancer, one in collaboration with Julian Pito and one with Paul Ferrer and Bruce Ponder. And those studies showed that BRCA1 and BRCA2 could account for perhaps 15 to 20% of the familial risk of breast cancer. So that was a little bit perplexing, and a question about what explains the rest. So as I said, we assumed that we could find further genes by linkage, and so we pursued that quite vigorously. But here there came across a twist. So this is a family that was, it's actually from the Netherlands, collected by Hannah Myers, and it looks for all the world like a high-risk breast cancer family, but it isn't linked to BRCA1 or BRCA2, despite the fact it's got a very large number of breast cancer cases in it. It turns out, though, that a lot of the cases do have are segregating a haplotype on chromosome 22, and somebody had the bright idea that actually chromosome 22 harbors the CHEK2 gene, another DNA repair gene. So perhaps we should sequence it. And indeed, it turned out that the individuals with this haplotype carry a truncating mutation in CHEK2, the now famous 1100 del C. So that was interesting, but what was surprising was that mutation was also present in the general population at quite a significant frequency. About 1% of the population carried it. And the mutation was only associated with a risk of about 2-fold for breast cancer. So that's much less than for BRCA1 and BRCA2. So now we have a breast cancer gene, but it's a completely different type of gene, conferring where the variants confer a much lower risk. So just digressing a little bit to the present, we can now because sequencing has moved on a long way, and we can now sequence on a much larger scale. It's been possible to define a much larger set of these so-called low penetrance breast cancer genes through so-called panel testing. So this is just to present the results from our own recent study, the so-called Bridges study, which has about 100,000 individuals sequenced for about 35 genes. And in this study, we're able to identify about 10 genes which are definitively associated with breast cancer. There's BRCA1 and BRCA2 at the top, TEC2, and 7 or 8 other genes with quite clear breast cancer associations, often subtype specific. And we're also able to show that a lot of other suggested genes, some of which are on panels, don't appear to be associated with. That's an important outcome in terms of being able to define rational panels for genetic testing. So, however, even if you take BRCA1 and BRCA2 together with these other genes, we can still only explain perhaps a quarter of the familial risk of breast cancer. So still the question remains, what explains the rest? This sets out what we know about the genes to date, and what we're summarizing here for each of the genes is the risk, so the relative risk of breast cancer on the y-axis, and the frequency in the population. So here we have BRCA1 and 2 up the top here, which were the ones found by linkage. And then we have these other genes, most of which they're rarer and confer lower risk. So one of the big questions is, could there be commoner variants in the population that confer increased risks? Are there, and how could we go about finding them? So I'm showing here actually the program from the BCLC meeting back in 1998, which is where that picture was taken. This was just to show that at the time we were thinking about the possibility that there were low risk or modifier genes. Unfortunately, at the time, we didn't really have the technology to tackle it efficiently because we couldn't do large-scale genome-wide association studies. We had to kind of guess the candidate genes, and that turned out to be spectacularly unsuccessful. Incidentally, I just noticed on the program that down the bottom you can see Kate Nathanson was also involved, which provides rather a nice link to the work of the Basser Center today. So it was clear that if we were going to be successful at finding common low penetrance genes, we needed a different strategy. It was no use using families because the risks weren't going to be large enough to give rise to those families. We had to use large series of women with breast cancer and controls. And we also needed a better technology to be able to type common variants across the whole genome. Unfortunately, that technology became available in the mid-2000s with these chip arrays, these SNP arrays, and it became cheap enough that you could type them on a large scale, and that's really the basis of the GWAS revolution. So this was our first success. It happens to be a marker near the FGFR2 gene. It's a 2-allele marker with 2 alleles, a G and an A allele, and you can see quite easily that the A allele is more common in cases and controls. You notice this was a very small study by today's standards, less than 400 cases and 400 controls, but the results look quite promising, and indeed, fortunately, it did replicate in a large number of studies, and it's subsequently been replicated many times. And this variant is reliably associated with breast cancer risk in all populations. Now, things have moved on a long way since then. The studies have got much bigger. That's largely through the formation of these two: Breast Cancer Association Consortium, the BCAC, the SIMBA Consortium that you'll hear much more about in BRCA1 and BRCA2 areas. And so now we have studies of well over 200,000 individuals, and the chips have also got better. So most of the data come from these two studies, the iCOGS array and the Oncoarray. And through these studies, we've been able to identify a lot more of these common low-risk variants. There are now more than 200. The results are illustrated in these Manhattan plots. So on the left, you have the Manhattan plot for overall breast cancer. And you can see these skyscrapers indicating the hits. So the FGFR2 is the biggest one, and a number of the other well-known hits, CHEK2 is there, TOX3, and so forth. On the right, you get the equivalent Manhattan plot for ER-negative breast cancer, and you can see immediately that the pattern is completely different. The genetic architecture for ER-negative breast cancer is quite different from ER-positive breast cancer. So that's important. There's this important subtype specificity, and it turns out that this goes a lot deeper. So this is some more recent work in the BCAC, led by Thomas Ahern and Monty Garcia-Clothas, looking at the subtype specificity of these associations. And so there's a whole series of different patterns. There's a class of variants exemplified by that FGFR2 locus that are predominantly associated with luminal breast cancer. There's another subset that's predominantly associated with triple-negative breast cancer. And then there's another subset of loci which appears to be associated more or less equally with all subtypes of breast cancer. The other main thing which we've learned is that most of these associations are not actually in genes at all. They mostly occur in regions which appear to regulate genes. So to the extent that we've been able to identify the causal variants, they appear to regulate nearby genes by regulating their expression. And so that's really led to a whole new class of susceptibility genes, not coding variants, but regulatory variants. This is from the most recent analysis of the BCAC data. In this study was published earlier this year, led by Laura Fakal and Hugo Ashard and Jonathan Beasley. These analyses showed that you could identify the likely target genes, over 170 likely target genes, some of which were known to be involved in breast cancer through tumor studies, but most not. So here we have a whole new potential class of genes that may be involved in breast cancer biology and potentially therapeutic targets as well. But how might one use these common variants in risk prediction? So individually, the variants don't confer enough of a risk to really be important. But the key point is that there's a lot of them, and so you can add their effects up. And that's what's illustrated here. So you have a distribution in the population of the number of risk alleles that somebody carries. So nobody carries all of them or none of them. Most people are somewhere in between. But breast cancer cases carry more than the general population, the basis of using these common risk alleles in risk prediction. So formally, how we do that is by combining into what's called a polygenic risk score, which is simply a sum of the number of alleles that somebody carries weighted by their effect size. That's what we did, using again the basic data from the BCAC. So we have a training set in which we tried out a whole series of approaches to developing polygenic risk scores based on different numbers of markers. And then we tested these out in a validation set, and then ultimately in a whole series of prospective studies. And what we showed was this was our favorite PRS, which is based on 313 SNPs. And this validates very well in all all prospective studies. So if you can look down all the prospective studies, the effect size is essentially the same, with an effect size, a relative risk of about 1.6 per standard deviation of the PRS. So a very reliable marker of risk. What does that mean in terms of absolute risk and in practice? That's illustrated here. So on the left, you have the lifetime absolute risk of breast cancer according to what percentile you are on the risk distribution. So you can see that there's a huge range, so that the women in the top 1% of the risk distribution have a risk of more than 30%. So that would put those women in the high risk category by any criterion. And at the bottom, the women in the bottom 1% have a lifetime risk of less than 3%. So there's a more than 10-fold difference where you are on the PRS distribution. On the right, what's shown is the 10-year absolute risk, and the horizontal line is a risk of 2.7%. So that's approximately the risk of breast cancer in women aged 50 in the UK, which is the age at which breast cancer screening is usually starts. What you can see is that the women in the highest risk category reach that level of risk at a much younger age. So in fact, in the top 1%, they reach that risk by about age 30, suggesting that screening in those women might be affected at a much younger age. Whereas women in the in the lower centiles never reach that level, suggesting that screening is perhaps they perhaps have too much screening, and perhaps screening could be reduced in those women. So this is really the basis of the ongoing programs to evaluate the acceptability of risk-adapted screening using PRS. One interesting thing which has been observed very recently is that the PRS also predicts the risk in other situations. So in particular, the PRS predicts the risk of a second contralateral breast cancer. That's been shown in work using the BCAC data led by Iris Kramer and Marika Schmidt. It also predicts for the risk of breast cancer in non-European populations, particularly in East Asians, the risk of male breast cancer, and as Antonis will talk about, in a number of other situations. So where are we now? We have really three categories where we have BRCA1 and 2, which still really stand on their own. We have this list of moderate penetrance, other genes that we can now use on gene panels. And then we have this large set of common risk variants, several hundred together. And what have we explained? Well, we think we've probably explained maybe 40% of the familial risk of breast cancer to date. Genes and regulatory SNPs. What explains the rest? Well, perhaps about 20% we think is due to other common variants that we haven't yet found because the GWAS haven't been large enough, and so larger GWAS may enable us to detect them. The remaining 40% is a bit more obscure. Maybe it's due to rare and non-coding variants or repeat polymorphisms or other things we haven't thought of, but that's really more for the future. So to finish off, I'd like to emphasize again that this is very much a collaborative effort. Hundreds of researchers have been involved in these studies. Some of the papers have 1,000 authors. Also, hundreds of thousands of patients, of course, have been involved in these studies, without which none of them would have been possible. Here are just some of the key people, either from my own group, other people in Cambridge, key collaborators throughout the world, and I thank them all. So thank you very much.
Thank you very much, Susan. So I would like to start, first of all, by thanking the Basser Center and the donors, Cherie and Len Potter and Mindy and John Gray and Susan Domcher, the director of the center, for this great honor as well. As Doug has said, the center has led really the efforts in supporting BRCA1 and BRCA2 related research since it was established. And I'm greatly honored and delighted to be in the company of previous awardees as well. But for me, the greatest honor is to share this award also with Doug Easton. When I first came to Cambridge about 24 years ago to study for my master's in statistics, I thought that I had all my career plans in place, that I'd be working in the insurance industry. But that year I chose to do a thesis in epidemiology, thinking that I'll be doing something different compared to what I'd be doing for the rest of my life. And then Doug was my mentor. He gave me the opportunity to work on some of the emerging data from the Breast Cancer Linkage Consortium at the time on the pathology of BRCA1 and BRCA2 tumors. And I guess I never looked back since then. So my journey here has been a combination of serendipity, but also, most importantly, inspirational mentorship from Doug during those formative years. So I'm immensely honored to be sharing this with him. Also, I would like to acknowledge at the outset all our colleagues and team members over the number of years here at Cambridge, but also all our collaborators from all over the world and the study participants. Without all this, the work that we do wouldn't have been possible. So back to the presentation. So women and men with newly identified BRCA1 and BRCA2 mutations face a number of key questions. So these include: what are the risks of developing cancer over time? What other factors may influence cancer risks? What are the implications for my close relatives? What is the optimal timing for the various interventions? What is the risk of the cancer recurring or developing second cancers? But also, what alternative methods of monitoring the disease may exist? So answering these questions have formed the motivation behind the work that we have been doing over a number of years, with the primary objectives of preventing and detecting early the BRCA1 and BRCA2 related cancers. So when I was talking to Susan about the subject of the presentation, she suggested that I give an overview of the work that we have been doing through the various consortia on characterizing cancer risks for BRCA1 and 2 carriers. So I'll try to summarize some of the main studies that we have been involved over time, and then also review some of the latest evidence on other factors, genetic, lifestyle, hormonal, that modify cancer risks for mutational carriers, and finish with some of the efforts to combine all this information into multifactorial models to provide more personalized cancer risks for BRCA1 and 2 carriers. So as I mentioned already, in the initial years after the identification of BRCA1 and BRCA2, due to the fact that mutations were rare and the technologies available at the time, large-scale case control studies were not really feasible in order to get precise estimates of the risks associated with BRCA1 and BRCA2 mutations. So most of the studies focus on the retrospective family-based design, where usually mutation screening takes place in affected individuals, and then we use those with mutations to collect additional data on family members. And over the years, we have been developing methods for estimating cancer risks using such family-based approaches while adjusting for the ascertainment. So the great opportunity came up in early 2000 to estimate the cancer risks in a large collaborative study. And for the first time, this was a study of risks in relatives of unselected breast and ovarian cancer patients selected for cancer family history. So here, through this collaborative effort, more than 8,000 patients were screened for BRCA1 and BRCA2 mutations, and for about 500 of those who had the mutations, we had family data, and then we used all this information to estimate age-specific risks of developing breast and ovarian cancer. So in addition to providing for the first time precise cancer risks using this large data set, this analysis provided a number of other important insights, in particular, for example, that the age-specific relative risk patterns for breast cancer are different between BRCA1 and BRCA2 carriers, that BRCA2 mutations are primarily associated with lower ovarian cancer risks compared to BRCA2, but also they demonstrated that the risks now in these families, which were identified through patients unselected for cancer family history, were lower compared to the risk that had been previously estimated on the basis of multiple case families. There's also evidence of the cancer risks varying depending on the cancer that was identified in the index individual, on whether, for example, it was a young onset breast cancer or older age breast cancer, whether it was breast or ovarian cancer. And the last three observations pointed to the fact that other familial factors may modify the cancer risks, which of course formed some of the hypotheses of future studies later on. Another observation was that cancer risks tended to increase with more recent birth cohort. Again, here one possible explanation being that some of the lifestyle and hormonal risk factors which influence cancer risks in the general population and which have been changing over time in the population may modify the cancer risks in mutation carriers as well. However, estimates coming from such studies may be associated with biases because in such family-based designs rely on self-reported cancer family histories, so which may be associated, for example, with inaccuracies, but also biases related to the way those families had been ascertained. So the ideal study design are prospective studies where usually we start with unaffected mutation carriers and then follow them over time to estimate a cancer risk. So such studies provide in principle estimates which are free of these reporting and ascertainment biases. But the main challenge here is that in order to get precise risk estimates, we really need large numbers of BRCA1 and BRCA2 carriers with long periods of observation. So we require really kind of the long-term investment. And a great example of the long-term investment done by Cancer Research UK is the EMBRACE study in the UK, the UK and Ireland, which recruits women and men with BRCA1 and BRCA2 mutations and also their family members. As I said, the study started recruiting back in 1998. So far, there are more than 12,000 study participants. So all the study participants provide detailed pedigree data at the time of entering the study, blood samples, detailed pedigree data, and there are then periodic follow-up questionnaires and also blood samples. We also obtain linkages with cancer registries to obtain information on incident cancers during the follow-up period, and we have access also to the medical notes to get information on pathology, surgery, and treatment. And more recently, through a grant funded by the Gray Foundation and led by Kate Nathanson, we'll be also collecting data on tumor blocks, so this is becoming a valuable resource. So just to give a couple of examples of recent studies that we have done on the basis of the EMBRACE prospective data. So BRCA1 and BRCA2 mutations had been associated previously with prostate cancer risks as well. But most of the studies have been retrospective and small in terms of sample size and with some conflicting results. So we use the prospective data on male carriers from EMBRACE to answer this question. So what we saw is that in BRCA2 carriers, there's about a fourfold increased risk of developing prostate cancer, which equates to around a 60% absolute risk of developing the disease by age 85. And there was evidence also that the associations were stronger with higher grade disease. but in BRCA2 carriers. Then there was also some evidence that the risks vary, for example, depending on the strength of prostate cancer family history in first or second degree relatives, or depending on where the mutation is located within the BRCA2 gene. For BRCA1, the associations were less clear and they were more sensitive to assumptions about screening in the cohort, but in the overall analysis, there was a suggestion of a twofold increased risk of developing the disease. We also used the EMBRACE data to answer the question on whether relatives of BRCA1 and BRCA2 carriers who themselves do not carry the mutations, so predictive test negatives, were at elevated risk of developing breast or ovarian cancer. So again, using the prospective data from EMBRACE. And what we see is that there's not really evidence of an increased risk. The relative risks when comparing the risks in those women were close to 1, suggesting they have risks close to the population risk. Of course, on the basis of the confidence intervals, we cannot rule out some modest increases, but to narrow down those confidence intervals, we need much larger samples. However, the major breakthroughs in the risk characterization of BRCA1 and BRCA2 came from the large international studies, and in particular two, the IBCCS, which is a collection of prospective studies with similar designs as EMBRACE. It currently has more than 50,000 BRCA1 and BRCA2 carriers with prospective follow-up, and focuses primarily on assessing the role of lifestyle risk factors on modifying risk, but also in estimating the risks of developing cancer prospectively. And the second consortium is the SIMBA consortium, which focuses primarily on genetic modifiers of risks. It has accumulated data now on more than 85,000 BRCA1 carriers from all over the world. So these consortia have accumulated now enough numbers that are allowing us to answer some of the key questions that I posed earlier with greater precision. So, for example, recently we used the data from the IBCCS study, but also in combination with data from the US, from BCFR, and from Australia, KCONFAB, to estimate prospectively the risks of developing breast or ovarian cancer in female carriers. So these results provided confirmation of the patterns that we saw previously in retrospective family-based studies, which of course it was reassuring that the risks that had been used were valid, but also kind of greater precision in those risk estimates. And they provided also some additional insights for the first time in a prospective setting. For example, clearly showing that the risks of developing cancer in BRCA1 and BRCA2 carriers vary depending on cancer family history as well. That risks increase with increasing number of affected relatives, suggesting, of course, that we should be taking into account cancer family history in the counseling process. Some of the original findings from the Breast Cancer Linkage Consortium was also that cancer risks in BRCA1 and BRCA2 carriers could vary depending on the mutation location within the gene. But those original studies were based on a few hundred study participants. So more recently, using the data from SIMBA with several thousands of mutation carriers now, we're able to look at this in more detail. So SIMBA suggested, the SIMBA analysis from SIMBA led by Kate Netherson and Tim Rebeck suggested that there are several regions in both BRCA1 and BRCA2 which may be associated with different risks of breast or ovarian cancer. And more recently, we're able to confirm this in the prospective data from IBCCS and actually estimate the absolute risks of developing cancer over time. So this is an example in the case of BRCA2, where we see clearly now that mutations which are located outside the central region of the gene are associated with around a twofold increased risk of developing the disease compared to mutations in the central region of BRCA2, and with similar findings also in BRCA1 carriers. Another observation from the original analysis of the Breast Cancer Linkage Consortium was that breast cancer in BRCA1 and BRCA2 carriers vary in terms of the histopathological characteristics. But again, those are based on a few hundreds of tumors at a time, and more recently using data from SIMBA, we're able to assess the distributions, for example, of estrogen receptors in greater detail to get age-specific distributions and clearly contrast the patterns between BRCA1 and BRCA2 carriers, which of course has implication on whom we end up screening for BRCA1 and BRCA2. But for us, they're providing the critical input parameters for risk prediction models and are now used routinely also in the BRCA1 and BRCA2 variant classification, for example, on the basis of the Enigma rules on deciding whether a variant is pathogenic or not. I think one of the key unanswered questions currently is what other cancer risks are associated with BRCA1 and BRCA2 mutations. Again, there have been several studies published. Most of them have been small in terms of sample sizes, and there have been some conflicting results. So what we are doing currently in SIMBA, we're using the pedigree data that we have several thousands of those now, and we are trying to assess the associations more systematically with 24 cancers with BRCA1 and BRCA2 mutations. So that's other than breast and ovarian cancer for females. So I have some very preliminary results here, so please treat those with caution at the moment. So we are seeing, for example, in BRCA1 carriers, associations with 5 different cancers, whereas in BRCA2 carriers, we see associations with 7 different cancers. There is some overlap, but there is also distinctions in the types of the cancer sites that are associated with BRCA1 and 2 mutations. So once these are finalized, we hope, of course, to obtain age and sex-specific absolute risks, which hopefully can form as part of recommendations for surveillance in men and women with BRCA1 and 2 mutations. So moving now on to genetic factors that may influence BRCA1 and BRCA2, that's other than the mutations, of course. So the first robust evidence that other genetic risk modifiers may exist came from studies of SIMBA, which have looked at the associations of the common genetic variants that were identified through the genome-wide association studies that Doug has just mentioned. So here there was robust evidence statistically to say, yes, these are modifying risks in mutation carriers as well. There was also, in 2010, the first GWAS published specifically in BRCA1 carriers, which identified a locus modifying breast cancer risk for BRCA1 carriers as well, but also there was a suggestion that it influences risk of estrogen receptor negative breast cancer in the general population. But most of our understanding in terms of the patterns of association of these other genetic modifiers came also by the participation of SIMBA within the iCOGS and Oncoarray experiments that Doug introduced already. So just to summarize what we see, if we, for example, take all the known breast cancer genetic susceptibility variants identified to date, it turns out that the majority of those appear to be modifying breast cancer risk for mutation carriers as well, but with some important distinctions. In BRCA1 carriers, the modifiers are the variants which are associated with estrogen receptor negative breast cancer in the general population primarily, whereas in BRCA2 carriers, the modifiers are those variants which are associated with the risk of overall or estrogen receptor positive breast cancer in the population. So in line with the distributions of estrogen receptor status in BRCA1 and BRCA2 associated tumors. One of the more recent analyses that has assessed the effect sizes of those genetic modifiers in BRCA1 and BRCA2 carriers, of all those known breast cancer susceptibility variants identified so far, has shown that for the majority, the effect sizes that we see in BRCA1 and 2 carriers tend to be attenuated compared to those seen in the general population, something that I'll come back to in a minute. When it comes to ovarian risk modifiers, what we see is that the GWASs that identified SNPs for high-grade serous, those SNPs appear to be modifying the risk of ovarian cancer in BRCA1 and 2 carriers as well. So what does this mean now for risk prediction? As Doug said, individually those SNPs are associated with small effect sizes, so they don't have great predictive ability on their own, but we get bigger effects once we start combining the effects together. So the most recent analysis from SIMBA has looked at the latest polygenic scores that Doug mentioned that have been developed in the general population and which have been optimized for predicting different disease subtypes. And we assessed how they predict the risks in BRCA1 and 2 carriers as well. So what we see is that the most predictive score in BRCA1 carriers is that for their negative breast cancer, whereas the most predictive scores for BRCA1 and 2 carriers, for BRCA2 carriers are the overall breast cancer PRS and the ER positive PRS that were developed in the general population. One important finding here in this latest analysis is that the hazard ratios that we see in both BRCA1 and BRCA2 carriers tended to be attenuated, smaller than the effect size that we see in the general population. So we try to investigate this further. So this could potentially result by the fact that BRCA1 and BRCA2 carriers are already at elevated risk of developing breast cancer. So those with high polygenic scores would tend to develop the disease at a young age, which would then imply a decreasing relative risk with age. And indeed, when we assess this, that's exactly what we see. We see significant trends in decreasing relative risk of the PRS with increasing age in both BRCA1 and 2 carriers. Another intriguing observation was that in both BRCA1 and BRCA2 carriers, we see differences in the effect sizes of the polygenic scores, but the predicted or the known effect of the mutation on the protein stability or expression. So more specifically, when we look at the effect sizes of the polygenic scores in pathogenic variants which result to a stable mutant protein, tend to be higher and more similar to those we see in the general population compared to the hazard ratios that we see for pathogenic variants which yield no or unstable proteins. And this, in turn, may suggest that some of the SNPs, for example, that are included in those PRSs may exert their effects through proteins which interact with wild type or mutant BRCA1 and BRCA2 proteins. So what I've shown so far was the effects of the PRSs in breast cancer, but we see similar patterns for ovarian cancer. And when it comes to male carriers, we see that the polygenic scores developed for females in the general population are also predictive of male breast cancer for BRCA1 carriers, but also the polygenic scores which are developed for prostate cancer in the general population are predictive of prostate cancer risk in also BRCA1 and 2 carriers as well. And in turn, these polygenic scores, as Doug said, mentioned for the general population, result in large differences in the absolute risks of developing cancer as well in mutation carriers here. So I'll try to demonstrate that here with an example in the case of BRCA2, somewhat simplified example where we know, for example, that the average risk for a BRCA2 carrier of developing the breast cancer by age 8 is around 68%. But then if we look at the BRCA2 carriers with low polygenic scores, their risk is predicted to be lower, around 48%, whereas for those who have high polygenic scores, their risk is predicted to be around 86%. So in this example, I'm using those who are at the 5th and 95th percentiles of the polygenic risk score distribution. So kind of in simple terms, kind of what the polygenic score appears to be doing is acting like volume control to moderate the risk of developing cancer. And where it makes even more difference is once we start combining these polygenic scores together with information, for example, on mutation location or family history, where the differences become even bigger. So a few things on lifestyle and hormonal risk factors. So as I said earlier, this has been the subject of study of the IBCCS study, and more recently, it has been reporting on the latest prospective analysis in collaboration with the BCFR and KCONFAB studies. So these are the largest prospective studies of their kind, trying to understand the role of various lifestyle, hormonal, and reproductive factors in BRCA1 and BRCA2 carriers. There have been also some alternative study designs like Mendelian randomization studies that were trying to answer these questions. Of course, I don't have really the time here to get into any detail. I tried to summarize what those latest analyses have shown, whether there is an association or not with risk for BRCA1 or BRCA2 carriers in these tables here. But of course, the underlying picture is somewhat more complex. We see differences in the effects of these lifestyle or hormonal factors in BRCA1 versus BRCA2 carriers, but also when we compare those effects to those known in the general population. There are also some inconsistencies between retrospective and prospective studies so far. So I think kind of one of the key messages here is that we really need to keep investing in these large-scale prospective studies to fully understand the role of several of those risk factors. So finally, a major focus of our work over a number of years has been the development of the Bodicea model, and to combine all this information on the various risk factors together. So the latest version of the model was published last year, which now includes the explicit effects of genetic variants in 5 different genes, including BRCA1 and BRCA2, the polygenic risk scores, family history, and also the lifestyle and hormonal risk factors. So this can be used in combination now to provide more personalized risks of developing breast or ovarian cancer. So, and some of our current work involves assessing the clinical validity of this model in independent prospective cohorts. And some of the initial results that are coming through those studies are encouraging. I have some of the, one example here where we have assessed Bodicea in the KARMA prospective screening cohort. These are data coming from Sweden, where we assess, for example, 2 versions of the model, one which includes the family history, the lifestyle factors, mammographic density, the polygenic scores, and then on top of that, adding gene panel testing information, which includes BRCA1 and BRCA2 screening as well. And what we see in both cases is that the predicted risks appear to be close to the observed risk, the 5-year predicted risks, so suggesting well calibration for the model in different deciles of predicted risk. And also that there is adequate discrimination between affected and unaffected individuals, suggesting that kind of this is a valid tool that can now be used for a risk prediction. Earlier this year, and I think Jackie mentioned that earlier in the previous session, we released also a new tool, which we call the CanRisk tool, and which implements this later version of Bodicea. So this allows healthcare professionals to enter information on the various lifestyle and hormonal risk factors, information on genetics, including polygenic risk scores, to build pedigrees online. And then the tool will produce the risks of developing breast or ovarian cancer or mutation carrier probabilities in an easy to communicate format as well. So for the last couple of slides, I wanted to mention a couple of reflections of what we are doing next. I think kind of there have been significant advances now in understanding how kind of those risks in BRCA1 and BRCA2 mutation carriers can vary. So we should be moving towards the implementation studies. So over in the last month, we heard that a new program has been funded through the International Alliance for Cancer Early Detection, which aims to perform now a randomized controlled trial for providing more personalized cancer risks for BRCA1 and 2 mutation carriers. So to assess the feasibility and acceptability of doing that, but also what difference it makes on the uptake of different interventions. And this will complement some of the other ongoing risk-stratified implementation studies, either at screening or at clinical genetics level, which are not necessarily focusing on BRCA1 and BRCA2 carriers. And I think we still have a number of questions to answer when it comes to studying risks for BRCA1 and BRCA2 carriers. So the aim is to capitalize on the resources that I mentioned earlier, but also new resources that are becoming now available, like linked electronic health records, to answer a number of those questions. So just to mention a couple, like understanding the cancer risks, for example, in cancer risks in carriers from diverse ethnic backgrounds, the long-term health outcomes of carrying BRCA1 and BRCA2 pathogenic variants, but also there's some exciting studies now using the EMBRACE data in trying to identify novel markers for early detection of cancer in BRCA1 and BRCA2 carriers. So that I would like to thank again the Basser Center, the Gray Foundation for this great honor, all our team members and colleagues and collaborators. So thank you very much.
We're going to start off with, are you planning to include the pathogenic variant location in the CAN-RISK model? And do you think it will substantially modify breast ovarian cancer estimated risk?
Yes, so it's one of our future plans. Yeah, it's on our list, to-do list, definitely.
Do you think it'll make? I mean, it's hard to, and I'll ask sort of a follow-up question. You know, the family history and the SNPs and the gene location. You know, how obviously they're kind of interrelated to each other, right? Because a family will have the same specific gene and location. So how do you make sure you're not double counting? I know it's going to be a stats answer, but do you understand my question? Like if you had
I guess when you get those estimates in the analysis, you adjust for each other, you know, whether, you know, kind of the data come from the same family or not. So I guess kind of when we come to add those kind of the effects within the model, you know, kind of we will appropriately adjust for those, you know, kind of effects. So those come out of the analysis in effect.
Great. I'll direct this to Dr. Easton. So do you think there are any other new, moderate, or high penetrant susceptibility genes left to discover?
So high risk, I think that's very unlikely. Moderate risk is still possible because only really the DNA repair pathways have been studied in great detail. So I think there's a new generation of exome sequencing studies which are being done now. I think we'll know, over the next 2 or 3 years whether there are any other significant players. But it looks like the genes that we've presented, particularly the 5 genes that are in Vodacea and the sort of 4 or 5 others that are known, they're the important ones.
And then we have a series of questions related to polygenic risk score modification of BRCA1 and 2 versus modification in moderate penetrance genes, and where will we have the greatest impact? And one of the comments is, and I think that this is the ongoing work that you and we and others are doing, which is how much does the polygenic risk factor change people's minds if they have a BRCA1 or 2 mutation, versus what the effect could be on CHEK2. So Antonis, obviously you published on this, and do you want to make a comment on that issue?
Yes, well, what we see is that clearly the polygenic scores influence the risk in both areas of the high risk variants, but also the moderate risk variants, where it makes perhaps more difference in terms of shifting on its own, in terms of shifting women into different risk categories are in the case of the moderate risk variants, where some of them can be shifted into the low risk or high risk categories. I think when it comes to BRCA1 and BRCA2 carriers, where it's likely to make the bigger difference is when we look at the age-specific risks at younger ages and deciding kind of on the timing of the various interventions as well. So.
Great, that's really terrific. Those are both fantastic talks, incredibly insightful and informative.